In fact, it was even better because the previous speaker has made an excellent introduction into the field of cosmetics and into the use of clays and other materials as parts of cosmetic applications. And most of my talk will be focused on the same topic. And I will speak about using of halloysite nanoclay for hair surface engineering. So generally this will also be focused on cosmetics and partially onto some topical drug delivery. But before I continue with the scientific part of my talk, I would like to thank those people who were involved into this study. This study was performed in 3 different locations. So this is Louisiana Tech University and the team led by Professor Yuri Lvov. This is the University of Palermo and team led by Professor Giuseppe Lazzara. And these are the members of my team in Kazan Federal University. So these people collectively have been involved into this study. This report is made based on 3 already published papers. So halloysite. Why it is important and interesting to work with halloysite. It is a natural clay, quite abundant, not as abundant as, for example, kaolinite, but still there are many deposits worldwide. And this clay is quite safe. So not much toxic effects can be attributed to this material. It has quite interesting morphology. So these are the tubes which have lumen inside, and these tubes, we always draw them like tubes, but in fact they might be not just tubes, but they might have rectangular shape, planar shape, some irregular shapes, but the most common thing is they are sort of rolled sheets of kaolinite, and they can be loaded by different components, by different compounds. So here you can see the lumen of halloysite nanotube, and the surface chemistry of this material is also quite remarkable. So it has alumina and silica layers, which makes them differentially charged. So they have negative charge on the outer surfaces and positive charge in the lumen, which helps us to load oppositely charged compounds either inside the tubes or onto the surface of the tubes. We have been using this material quite extensively, and my first induction into the use of halloysite nanotubes was related to the topic which we also have been working on, which we term cell surface engineering. So in this case, we engineer the surfaces of cells by applying different types of materials onto these surfaces. So here, for example, you can see yeast cells, which were coated with polyelectrolytes, and between the layers of polyelectrolytes, we introduced halloysite nanotubes. So this is a kind of mixture of several different techniques. We have layer by layer assembly, we have inorganic nanotubes, and we have cells. And we can, for example, remove these cells by burning them, and we can obtain whole of halloysite, or we can load some functional components, like, for example, we can load nutrients into these tubes, and this will provide the cells with some additional food. So this will help the cells to grow, or we can modify other properties of the cells. So that's what we have been doing with biological cells. And at some point, we had an idea, why not using the same approach to modify the surfaces of hair? Basically, hair is very similar to cells in the first approximation, because actually it is also the product which is produced by cells. So we have cells which produce this keratin, this protein, and the hair fibers are the protrusions of the cells. So we can say that these are also cells or parts of cells. And they are built of protein, but in fact, the surface of hair is also coated by lipids. So again, in first approximation, the surface of hair is quite close to the surface of cells. We have lipids both in hair and in cells. And you can see here on these images that hair is quite rough. So it has several layers. And the outer layers, they produce some kind of tiles. And these tiles, they have some dimensions, and they have some areas where one tile is covering another tile, like scales in fish, for example. And this means that the surface of hair is not uniform. So we have some areas which are different from the other areas. Just to mention, all mammals, including humans, are covered to a certain extent with hair or fur. And there are some animals which lack hair, and humans actually also lack hair on the most part of their bodies, again, to a certain extent. And but in general, all mammals are covered by hair, and the same essentially applies to birds. They have feathers, but these feathers are more or less analogs of hair. They have a bit different structure, but they're also produced from the same keratin protein, which means that if we can produce a technology to modify the surfaces of hair, this technology will be readily available to mammals and birds, and of course to humans. And here I show some images which demonstrate the necessity to modify hair surfaces. So some people, for some reason, are not happy with gray hair, and they need some dyes, some means to color their hair, and this helps them to be more, let's say, to increase their self-esteem and to be happy with their outlook. So this only seems to be a sort of cosmetical problem, but in fact it might be a medical problem, because if somebody is not happy with their hair, they might have stress, they might have some mental conditions. So this means that coloring hair is not just cosmetical problem, it is also a medical problem. And the same applies for colored hair in cases that people are just not happy with their natural color of hair. We can also treat hair. For example, in this case, application of keratin protein to keratin on hair helps to make hair looking better. So you might increase the volume of hair, you might straighten your hair, you might protect hair from, let's say, ultraviolet and so on. And finally, let's say more medical application is tackling hair parasites. So this is applicable to humans and this is applicable to animals. So we can be infested by different sorts of parasites like fleas, lice, just to mention, and we need to get rid of them. We need to somehow protect ourselves. There are some medical formulations to do this, and these are applicable to humans, but when it comes to animals, it becomes more and more complicated for obvious reasons. We can wash our hair daily, animals cannot do this, especially when we speak about agricultural animals. But actually, these parasites, they significantly decrease weight of the animals. They can kill younglings and so on. So it is quite important to tackle hair parasites. So to do this, the team which I presented in the second slide proposed a peculiar approach, which is based on self-assembly of halloysite nanotubes on hair surfaces. So we found that if we wash hair with aqueous dispersions of halloysite at, for example, 1 weight %, this number can be either reduced or decreased or increased depending on what we want to get. This results in deposition of halloysite onto hair surfaces. So without any treatment of hair, without any treatment of halloysite, we can obtain quite robust and quite dense layers of halloysite on hair surfaces. So here you can see some microscopy images which demonstrate pristine hair, not coated by anything except natural lipids and dandruff in some cases if the hair is not washed. And here you can see the halloysite coated hair. And here you can see, for example, the distribution of silica and alumina on hair imaged using X-ray spectroscopy. We found that this coating is time dependent. So basically, if we hold hair for longer periods of time, this results in better coating. If we just immerse hair for short periods of time, predominantly halloysite is concentrated between these intercuticle voids. So as I told you, and I will show you a little bit later, these areas are quite different from the surface of the hair. So these areas are the targeting site for clays if we coat hair with clays. Obviously, concentration also is an important factor when we coat hair with halloysite. So if we apply more concentrated suspensions of halloysite, we will get a more dense surface coating of hair. So here, for example, you can see at 2 milligrams per milliliter, we also have halloysite mostly deposited in the voids, and with increasing concentration we have the whole hair shaft coated by halloysite. We also found an interesting feature that if we hydrophobize halloysite nanoclay, so if we coat halloysite with some hydrophobic materials, here we used organosilanes, this increases the coating efficiency. We attribute this to the fact that human hair and in general animal hair is intrinsically more or less hydrophobic. In some animals it is very hydrophobic, especially in aquatic and in semiaquatic animals. And in humans, and let's say in other non-aquatic animals, hair is relatively hydrophobic. This is needed to get rid of liquid. So if we are, imagine if you are on the seaside and if it is quite cold outside, we need to get wet immediately, otherwise we will get cold and so on. So if you don't have any towels, the only way to do so is to get rid of water naturally, and if the hair is more or less hydrophobic, the water moves out of hair faster. That is the reason for hair to be hydrophobic, the biological reason. And if we increase the hydrophobicity of clay, this provides us with more sites of deposition and results in very high density of clay coating. So here you can see that this also affects the robustness after washing. So if we wash hair with shampoo or rinse it with water, in case of pristine halloysite, it is being removed much more efficiently than in case of hydrophobic halloysite. So higher the hydrophobization, better is the deposition efficiency. And this works pretty well with most types of hair we tested. So here you can see dog hair, or fur, cat hair, horse hair. In all cases, we have very good coating, which can be numerically evaluated by, for example, measuring the roughness parameter before and after deposition of halloysite. And here, for example, on this slide, you can see how this approach can be utilized for staining human hair. So if you load commercially available dyes or natural dyes, which are in most cases hydrophobic, into halloysite nanotubes, this will help you to stain, to color the hair. And here, for example, some of you might recognize this volunteer, some might not. So here you can see this person has stained hair with henna-loaded halloysite, and in this case there is no signs of allergy. So in case of direct application of the dye, you can get this allergic signs, but in case of dye loaded into halloysite, there was no allergy, which we attribute to the fact that the dye is loaded into the lumen of halloysite. It has been washed out of the surface of halloysite, and that's why we do not have that intensive allergic reaction. Another application of this technology is photoprotection of hair. So as many of us already know, keratin, hydrolyzed keratin, is used in some hair care products to treat hair. And we were thinking, why not loading keratin into halloysite nanotubes and then apply halloysite nanotubes loaded with keratin onto hair. So replace pure keratin particles with keratin absorbed by halloysite. So here you can see that some physical chemical data on changes which are induced by loading of keratin into halloysite nanotubes. I will not stop much on this. Here you can see the images of atomic force microscopy images of halloysite nanotubes coated by keratin. So these are the keratin particles, and these are the halloysite nanotubes, which have keratin both outside and inside. We cannot say that all keratin is inside halloysite. Pretty much of it is on the surface. And you can see that stability of keratin is increased if we load halloysite with keratin. And on these images, you can see that the colloidal particles are quite stable and they do not coalesce. And if we apply this formulation onto hair, you can get essentially the same result as we obtained with pure or hydrophobic halloysite. And here you can see coating of hair for 15 minutes and for 60 minutes. Obviously, we aim at shorter periods of time because nobody would, somebody would wash hair for 60 minutes if that is needed. But normally we do not waste that much time for washing hair. And if we can reduce this time drastically, this will allow us to use this formulation commercially. So if we apply keratin and halloysite for 15 minutes, this results in quite a good coverage. And here you can see some numerical data on microscale texture parameters. So if we measure the roughness parameters of pristine hair and halloysite coated hair, we can see that some parameters are being changed. And some parameters which indicate the waviness of the hair as the whole, they are not really changed. This means that we can get some changes at microscale, but on, let's say, semi-macro scale, there are no changes. So hair will appear exactly as it is without this coating. We can actually attenuate the appearance of hair. So if we have more halloysite on the surface of hair, it becomes more reflective and it can reflect hair. So we can attenuate the hue of hair. And this happens without any dyes. So the hair becomes, so it is looking more shiny than the normal hair. Here you can see the deposition of halloysite nanotubes on hair surfaces. Again, depending on concentration, we either have the predominant deposition sites at the voids between the cuticles, or we can coat the whole shaft of hair with halloysite nanotubes. And here you can see some results on photoprotection. So if we irradiate hair with ultraviolet, this leads to photo degradation of hair, which can be detected by detection of cysteine, which is amino acid in keratin. And if we coat hair with halloysited keratin, this reduces the production of cysteine, which indicates that no photo degradation occurs. So this means that this coating protects hair from UV radiation. We do not know for sure which component is more important here, either halloysite or keratin, because we didn't study this differentially, but together they work quite good. And they also protect hair from mechanical degradation. So you can see that if we have pristine hair irradiated by UV, you can see this rough areas which indicate the degradation, the mechanical degradation. Here we don't have them because the hair is protected. And the same applies for mechanical properties. So as a function of mechanical properties of hair, we can estimate the degree of photo degradation. The final part of this talk will focus on anti-lice or anti-insecticidal properties of halloysite nanocontainers. So we first studied this with Caenorhabditis elegans, which is not insect at all, but it also can react on some poisonous compounds loaded in halloysite, and we found that this system works with C. elegans nematodes. But of course, we were more interested in lice, because lice are a very significant problem, even for humans. Although in developed countries, we normally do not have lice as we used to have them 500 years ago or something like this. But in some other countries, and especially in the areas where we have some natural disasters, we have these problems with lice infestation, and we need to develop some formulations which would have long-lasting anti-lice protection. And here we focused on some animals which are peculiar in their own right. So we have chosen guinea pigs and capybaras. Guinea pigs and capybaras are quite relatives in terms of evolution. So they are like cousins to each other. Although they live in completely different habitats. So capybaras are semiaquatic animals. They are actually the largest rodents, world's largest rodents. They are of average dog size, so like German shepherds. Some of them can get 70 kilograms in weight. Guinea pigs are much smaller, and they have hair. Both of them have hair, but hair of guinea pigs is quite similar to human hair. Its purpose is to protect, to thermo-insulate these animals, which actually live in natural habitats in mountains, so they need some protection from cold weather. While capybaras live in tropical areas, they have no problems with temperatures, but they have problems with water because they live in water and they need water to be removed as fast as possible when they get out of water. So here you can see that the hair of capybara is completely different. It is produced from two hair shafts, which are connected to each other with sort of wax-like, we call it wax, substance, which is very hydrophobic. So here you can see, for example, the electron microscopy images of hydrophobic capybara hair, and that's how pristine halloysite assembles on capybara hair. Here is the capybara near the pool, indicating that it is a semi-aquatic animal. So here you can see the side view of halloysite deposited on capybara hair. Guinea pigs, in turn, are not aquatic animals at all. They hate water. They don't like being exposed to water. And in their case, halloysite is predominantly located in the voids, which are apparently the most hydrophobic part of guinea pig hair. And the same applies actually to human hair as well. And here you can see some atomic force microscopy images of capybara hair and guinea pig hair. So you see on these mechanical mapping images that capybara hair, which is covered by this wax, is more or less uniform. So we do not have very soft areas. Whereas in guinea pig hair, we have very soft areas near the voids, which means that apparently softer lipid-rich areas are located here. That's why we have this predominant deposition of halloysite in the voids. As in case of humans, we can detect some mechanical and especially topographical changes in capybara and guinea pig hair, especially if we compare pristine capybara hair and capybara hair where we remove the wax. So we can remove this wax by acetone, for example, which allows us to get hair which is very similar to guinea pig hair. As I told previously, they are actually cousins. So at some point in evolution, they were the same animal. That's why if we remove the wax, the capybara hair is very similar to guinea pig hair, and this manifests in lower intensity of deposition of halloysite. So pristine capybara hair can be modified with halloysite much more effectively. And if we investigate the hydrophobic properties of pristine capybara hair, pristine guinea pig hair, and capybara hair treated by acetone, so less hydrophobic hair, you can see that here the capybara hair is most hydrophobic. That's why we have this effective deposition of halloysite. And next, we actually found that the deposition of halloysite results in approximately 5% weight increase if we deposit it onto hydrophobic capybara hair. And if we load halloysite with permethrin, which is a hydrophobic insecticide drug, this would help us to obtain nanocontainers, which are quite stable and which have a quite extended release of permethrin. Normally, permethrin is being deposited directly onto hair. So we wash hair with water which contains permethrin, which is very hydrophobic. That's why we do not have uniform coating, and that's why this coating is not robust. So it can be removed with washing hair. But if we put permethrin into halloysite, this provides us with very extended release. And here we moved yet to another animal, which are goats, which are agricultural animals. And we we checked if pre-treatment of goat hair with artificial wax changes the deposition efficiency, and it does change the deposition efficiency. So if we pre-treat goat hair with wax, it will get coated with halloysite exactly in the same way as natural capybara hair. And if we wash this hair, so this is the hair after deposition of halloysite without washing. Here it was washed once intensively, and here it was washed twice intensively. We need to keep in mind that normally agricultural animals are not washed. We don't need to wash them, but they can get washed, for example, under rains or while crossing rivers, something like this. But this washing is not that intensive as how we wash our hair in shower, for example. But still, in this case, we still have very efficient deposition of nanocontainers, which are loaded with insecticidal drugs. And then we checked how this works using lice. We have chosen chewing lice. So chewing lice are different from blood-sucking lice. They do not actually bite animals or humans, they just eat hair. That's why they are not that dangerous, but still they can be dangerous because they transmit diseases and so on. But biologically, they are the same as blood-sucking lice. And if this works with chewing lice, it will also work with, let's say, normal lice. And here you can see how we produce this experiment. So we used hair, coated with halloysite, loaded with permethrin, and we put lice onto this hair, and this is the live animal, this one is the dead animal. So you can see it's shrunken abdomen, lack of these legs and so on. So you can distinguish, and they don't move. That is the most important part. So you can distinguish dead animals from live animals. And here you can see that if we wash hair 2 times after exposure to pure permethrin, after 2 washings, we do not have any protective effect. So we can still record live lice on hair. But if we wash hair and this hair is treated with halloysite loaded with permethrin, we do not have live animals, so they all die. So this means that we have this long-lasting effect. And it is even better shown in this table. So here you can see that we exposed animals onto the hair, which was washed 2 times after 1 month. So after 1 month, we still do not have any live animals in the hair, which was treated with halloysite, which means that we can render these protective properties at least for 1 month, which is very important for agricultural animals, which can be moved to pastures, to natural habitats, if they are kept outdoors without any need to other treatments on farm. This might be important for pets. And this also technically can be applied for humans, especially for humans which do not have access to normal hygienic facilities, which happens during disasters like floods, earthquakes, and so on. And so these are some conclusions. We think that this can be extended to antifungal drugs, and also for some topical drugs which are used to treat skin diseases in humans. And before I finish, I would like to pay my tribute to these animals, which were the subject of this study. They have never met in person because this one lived in Texas and this one lived in our house in Tatarstan. They were both born in 2015, and unfortunately they both died this spring. But they contributed greatly to this research because only after I saw this animal, which was kept as a pet by one of my friends, I understood that there are some interesting similarities which might be manifested in the structure of hair. And here,
they died not because of your experiments.
No, absolutely not. The hair was collected without any, any damage to the animals. So they are brushed every day, and when you brush them, you just get this hair. And I would like to thank Melanie Tipaldas from Rawil Foundation for Capybara Veterinary Medicine, who actually provided the capybara hair, so you can appreciate the sizes of this animal. And I would like to thank Russian Science Foundation for funding this research, and thank you very much for your attention.
Thank you very much, Rawil. We are running just too late. And then, so I just, there is a short question.
I have one question, very short.
Okay, Jerry, go.
Rawil, 1 second. What
Sure.
you estimate from your experience in wet state, what is gap of open cuticle?
Do you mean the gap between 2
Opening.
cuticles, or the height between 2 cuticles?
Height to penetrate inside.
In wet state, it might reach 3 to 500 nanometers, depending on
Okay.
species.
I agree. Submicron,
Yes,
hundreds,
less than a micron.
hundreds nanometers.